Hello everyone, this is Dhanashri from the Dr. Omics Research Lab. In this session, we are going to discuss about the functional enrichment analysis. What is the meaning of functional enrichment? Now, the name itself, it is telling that which function is highly enriched in our data that we are going to find out with the help of functional enrichment analysis. Because up to now we have filtered the significant genes, we retrieve the annotation, but it's not like that one gene is only involved in one function. One gene can perform multiple functions. So which function is affecting in our data or like most of the genes from our list, they are involved in which function that we are going to find it out with the help of functional enrichment analysis. So here we just required two columns like the function which is highly enriched and the number of genes from our list which are involved in that. And this step we are going to perform separately for the upregulated and downregulated because function is depend upon the expression. And for that we are going to use the Panther database. But before that, let's filter out the upregulated and downregulated genes. This is what the significant genes we are having. And to filter out the upregulated and downregulated, we need to apply the filter on the log to fold change column. So the cutoff for the upregulated is greater than or equals to 1. So we are just applying the filter log to fold change number filter greater than or equals to 1. And this is what the filters we are getting. So I have already prepared the sheet for the upregulated. You can paste it. You can just create, rename it. Otherwise, already create it and then paste. Then same thing we are going to do for the downregulated. So for the downregulated, the cutoff is fold change should be less than or equals to -1. I'm using the standard cutoff. And here this is what the downregulated genes we are having. Now go back to the significant sheet and remove the filters so that you have your original data here and your filtered data in the new sheet. Now I'm going to show you for the upregulated. Same thing you can do for the downregulated as well. So we just have to open the Panther database and here you need to submit your gene list. So from the upregulated, I'm going to take the gene symbols and in this enter ID box, we are going to paste it. We just need to select the organism Homo sapien. If you have any another organism, you can check and select accordingly. We are going to represent this enrichment with the help of graph so that we can visualize it properly. And that's why here I'm selecting the option functional classification viewed in graphic chart and selecting either the bar chart or a pie chart. You can select as per your choice and submit. So you can see here it is showing the pie diagram and this is for the molecular function. If you will see in the molecular function, this purple color is taking the maximum area, but this is for the no Panther criteria is assigned, like there is a no molecular function for it. But after that, the red color is occupying the maximum area, which is for the binding function, which means the binding function is highly enriched in our data. Means most of the genes from our data, they are involved in the binding function. If you want to see which genes are there, you just have to click on the function and it will show you the function and the gene list which are involved in the binding. This is for the molecular function. If you want to see for the biological process, so here also you can see for the biological process, biological regulation is enriched. For the cellular components is also given. For the cellular component, cellular anatomical entities enriched. And here for the pathway also it's given, you can check it out. But whenever we are preparing a output, we need a Excel sheet Or you can say we need a column-wise information like the functions and the number of genes from our data which are involved in that function. So right now as we are visualizing the molecular function, for that you have to click here on the export, so which is going to export the result. So here it is showing the Panther chart, and this chart you can open with the help of Excel. Delimited it, tab separated only, next and finish. Now here if you will see it is showing some 5 columns. First column is the serial number that I'm going to delete, and last 2 columns is showing the percentage that also I'm going to delete because we required only the function and the number of genes from our list which are involved in that. So this is the function in the first column. This is a molecular function for upregulated gene, and this is the number of genes. If you want, you can just delete this no panther criteria assign. And now here as we have a function, as we have the number of genes, we can generate a pie chart. Either you can take a screenshot from the Panther database, otherwise you can generate and edit as per your choice. You just have to select the data, then insert. You can generate a bar chart also or pie chart also. And here there are different types of pie charts also we are having. You can select as per your choice. You can edit as per your choice. Then if you want, you can just change the title. You can just right click and add the data labels as well. And this is how you can edit the chart as per your choice. So this is for the molecular function, which is upregulated molecular function. Same thing you have to do for the biological process, cellular components, and for the downregulated also. Means it will be total 6 chart. But for the pathway enrichment analysis, you need to combine both the upregulated and downregulated genes. Otherwise, simply you can just copy the gene IDs from the significant sheet and then submit it into the Panther database to retrieve the pathway information. Because pathways are not depend upon the expression, so you can combiningly also generate the pathway enrichment by the same way which we have discussed in the Panther database. And this is about the functional enrichment analysis. So here this pie chart or a bar graph, we need to visualize the enrichment over here. That's it for the enrichment. If you have any question, let us know. Till then, bye. Have a great day.